Hello everyone, welcome to Microtalks. Today we will be discussing the Messelson and Stahl's experiment, which is a very famous experiment that proved that DNA replication is semi-conservative. In 1958, Matthew Messelson and Frank Stahl, they proved that DNA replication is semi-conservative. At the time, two other models were proposed to explain the DNA replication, that is the conservative model and the dispersive model. In the conservative model, the original DNA molecule remains intact and a new identical molecule is produced. As you can see here, this is the parental DNA molecule which remains intact here in the progeny, and an identical DNA molecule is produced. There was another model that explained DNA replication, that was the dispersive model. The new DNA here is a mixture of old and new segments that are interspersed along the strands. As you can see here, the blue is your old DNA and the red is the new DNA. So it gets interspersed along the strands along with the old DNA to form the new DNA strand, which is the progeny strand of the second generation. The semi-conservative model explained that each new DNA molecule consists of one original strand, as you can see here in blue, and one new strand in red. So it is a mixture of one parental strand and one newly synthesized strand. So now let us have a look at the experimental design of Messelson and Stahl. So what they did was they used E. coli, which was grown in a medium containing a heavy isotope of nitrogen, namely 15N, which is a heavy isotope. After several generations, all the DNA was labeled with 15N, thus making it dense, as you can see here. Then the cells were picked up and they were shifted to a lighter medium. They were transferred to a medium that contained the lighter isotope, that is 14N. As the bacteria replicated, new DNA strands incorporated 14 nitrogen, thus making them lighter. The DNA was then extracted, from each generation and subjected to density gradient centrifugation. They used cesium chloride density gradients to separate the DNA based on the density. So heavier DNA, which was there in the first generation, settles lower in the gradient. As you can see here, heavier DNA settles lower in the gradient, while the lighter DNA settles higher in the gradient. There was one more band that was found on the cesium chloride density gradient, and that band was that of the intermediate density. That indicated hybrid molecules having one strand of heavy nitrogen, that is 15 nitrogen, and one strand of 14 nitrogen, that is the light nitrogen. So this theory supported the semi-conservative model. After subsequent generations, the experiment yielded DNA population of both intermediate density and the light density, consistent again with the results of, or with the hypothesis of semi-conservative replication. Each Each new DNA strand contains one old strand and one new strand. So, in conclusion to the Messelson and Stahl's experiment, we can say that the results confirmed the semi-conservative model of DNA replication. The experiment conducted by them was pivotal in understanding the fundamental mechanism of genetic inheritance and DNA replication, and this experiment confirmed the Watson and Crick model of semi-conservative replication. Thank you all for watching my channel. Please do like, share and subscribe to my channel. Looking forward to bringing you many more episodes of Micro Talks. Thank you for watching.